My name is Tatiana Koshla, and together with our team at BinomLabs, we closely analyze the experimental research study entitled Targeted CD47 checkpoint blockade using a mesothelin-directed antibody construct for enhanced solid tumor-specific immunotherapy. First, I would like to express my respect to the researchers for this work. It is a strong and elegant study that combines careful molecular design with rigorous biochemical and cellular validation. This research focuses on a very important therapeutic challenges. How to achieve combine low affinity CD47 checkpoint blockade and specific tumor targeting within the multivalent and multifunction antibody construct, while reducing the toxicity commonly associated with the CD47 direct approaches. Let's move directly to the area of biochemical laboratory research for which the implementation of the AI platform will provide predictive results of experimental data. Because the BinomLabs software platform is designed to predict biochemical experimental outcomes related to binding affinity and interaction behavior within molecular complexes, this paper is especially interesting for us. Our platform is intended to support the analysis of systems where researchers need to understand or estimate physical and thermodynamic quantities such as affinity-related behavior, entropy-related changes, dissociating-related energetic trends, interaction energy between paired amino acid residues, the stability-related parameters relevant to molecular recognition. In the present work, we focus on the result that is closer to the predictive scope of BinomLabs and most directly connected to physical affinity measurements. On this slide, we highlighted the key result showing that the engineered mesothelin target antibody construct binds specifically to mesothelin and is also capable of blocking CD47. In the panel A, surface plasmon resonance sensogram stress the binding of the indicated antibodies to mesothelin on the left and in the middle, and to CD47 on the right. This is especially valuable because surface plasmon resonance methods is a direct physical method for measuring interaction behavior and affinity-related properties. From the BinomLabs perspective, this type of experiments is highly informative because it reflected a key dual design requirements. The construct must preserve selective tumor anchoring through Mesothelin while also maintain functional CD47 blockade. This is exactly the kind of multi-parameter molecule problem where predictive prioritization can be useful before extensive experimental screening. In a workflow, BinomLabs may help researchers in next task. Eliminate variants that are more likely to lose tumor target anchoring. Reduce the number of constructs that require for SPR and cell binding validation, surface plasmon resonance. Focus flow cytometry analyzing on variants most likely to preserve meaningful discrimination between mesothelin positive and mesothelin negative system. And rank candidate local inhibitory checkpoint monoclonal antibody, LICMAP, designed according to their likelihood preserving both Mesothelin-specific binding and functional CD47 blockade. Important experimentally, because a major part of the experimental cost in such project lies not only in the final validation, but in the many intermediate construct variations that may fail early at the level of binding selectivity, function blocking performance, or overall molecular balance. In this context, BinomLab may help identify before surface plasmon resonance and flow cytometry testing which local inhibitory checkpoint monoclonal antibody architectures are more likely to preserve strong and selectively mesothelin recognition, so thereby reducing low-value construct producing and unnecessary binding screens.
Don't lose your chance.
Our company provides biochemical laboratories with access to a modern molecular prediction platform that generates preliminary in silico experimental forecast for research questions involving antibody-antigen interaction, structural biology, protein-protein interaction, amyloid peptide aggregation, different models of inhibition, enzyme-ligand interaction, and related molecular systems. If you do a biochemistry experiments in the lab, take advantage of the free opportunity to predict experimental results. We built an AI platform that calculates a broad spectrum of thermodynamic parameters, including stability indicators, using modern computational and software testing on real molecules. In solution, we also integrate three-dimensional structure from PDB files, AlphaFold, molecular dynamic, and molecular kinetics. We use only the quality experimental input measurements, purified proteins, precise temperature control, and accurate salt or denaturant concentrations. Our key features is directly visual correlation between calculated and experimental values. For example, the correlation reaches about 90% between experimental and prognosis data.
Don't lose your chance.
In vitro research typically accounts for 20 or 40% of total R&D expenditures in the pharmaceutical industries, and improving prediction reduces waste cycles. General success rate for well-characterized interactions can improve, for example, from 50 to 75%, and from 70 to 90%. In a free pilot project on forecasting experimental data, if you are a biochemistry specialist in the lab, take advantage of a free opportunity to predict experimental results. Don't lose your chance.